Welcome to this video on how asexual prokaryotes achieve genetic diversity. Before I talk about prokaryotic cells, I'd like to first review what we know about eukaryotic cells. We know that most eukaryotic cells can divide both asexually and sexually. For sexual reproduction, this is going to involve meiosis, so that we can form haploid gametes. In sexual reproduction, most eukaryotic organisms will achieve great genetic diversity through a series of some crossing over events and independent assortments of individual chromosomes that will contribute to this great diversity within the population. What I'd like to show you is that these cells, these are the primary cells of a eukaryotic cell, they're going to be diploid. That means that they have 2 copies of every single chromosome. So both parent cells have double the amount of chromosomes. They're going to go through these 2 rounds of meiosis so that they can form their gametes, whether they are the eggs or the sperm, and these gametes are going to be haploid. When these gametes fuse, the whole point is to make a gamete that is back to being diploid. So we're going to acquire some genetic makeup from both parents. There's that diversity. Now this can only occur if the host cells have a diploid chromosome and form haploid gametes. So recall that fungal cells are haploid in nature, and so they need to undergo plasmogamy, karyogamy, and then these rounds of meiosis in order to achieve their genetic diversity. This slide basically shows you that flow of genetic diversity found in eukaryotic organisms. It can be seen as a vertical gene transfer when we set up a genealogy tree where we have the ancestors on the top and the current offsprings at the bottom. So we're going to go from top to bottom in this case. We can always reverse it, go bottom up, but we're going to go from top to bottom. When the gametes of 2 genetically distinct parents form an offspring, such as these 2, the new combination of genetic material from the diploid offsprings will then be unique from each other. That means that this great-grandchild number 1 has a unique genetic makeup compared to his parents. And this genetic diversity is going to be carried downwards from generation to generation. So when we eventually get to us, we'll carry some genetic material, but we will acquire others along the way. As I mentioned, that eukaryotic organisms also can undergo asexual reproduction. Not all, but some. In this case, there's still going to be some vertical gene transfer, but it's going to be of the asexual nature. The parent cell's genetic makeup is going to be transferred to their offspring in a nearly identical copy. That means that the daughter cells are going to be clones from each other. And they're going to do this through mitosis instead of meiosis. So now let's get back to prokaryotes. Prokaryotes are basically haploid. They have 1 chromosome and they only have 1 copy, so they are haploid to begin with. In the case of prokaryotes, such as bacteria and archaea, these organisms can only reproduce asexually, so their vertical gene transfer will only be that of asexual reproduction. Since there's only 1 parent cell and there's no exchange of genetic information, the daughter cells will have the same genetic makeup as their parent cells, and thus they're going to be clones of each other. The question is then, if prokaryotes can only pass their genetic makeup through asexual means via this form of vertical gene transfer, then how can they exchange DNA to achieve this genetic diversity that is often required to maintain a species? We do know that this exchange of DNA between bacterial cells does occur, and this exchange is going to be called horizontal gene transfer, because the DNA is acquired not from parent down to the offsprings, but instead from one organism to another. They offer neighboring organisms, and they're unrelated genetically. So, from this organism to this organism, we're going to transfer genes along the horizontal way. We also know that this horizontal gene transfer is going to occur in one of three ways: that of through transformation, transduction, and conjugation. In prokaryotic horizontal gene transfer, transformation is where naked or foreign DNA is going to enter from one host cell into another. So it's just pure DNA. That's all we need. I'll explain that further in the next few slides. Transduction is a transfer between cells with the use of a virus. And if you recall, the viruses that infect bacteria are bacteriophage. So we're going to need a bacteriophage in this case. For conjugation, we're going to need a pilus or an F pilus that is a hollow tube, and it's going to allow the transfer of genetic material from one cell to another. And so that's going to be conjugation. So I'm going to go into these three steps in a little bit more detail. Let's start with that of transformation. In transformation, the prokaryotes take up naked DNA, as I mentioned, so there must have been a neighboring cell in the area that died, and during the process of cell death, they will fragment their DNA. And so their DNA is now free in the environment. In nature, the bacterial transformation is an important mechanism for acquiring this genetic elements that encode virulence factors and antibiotic resistance genes. So if this bacteria that died had any toxin genes or virulence genes, it can be easily acquired by passing through the membrane of a neighboring cell. This process is actually very inefficient because the environmental DNA levels are very low to begin with, but also bacteria tend to have some enzymes that will digest foreign DNA. So because of that, this does occur, but it occurs in a very low rate. The earliest example that we have of transformation is in a mouse experiment where there was a non-encapsulated bacteria, that's a bacteria without a capsule, was shown to acquire a virulence gene that now allowed it to produce this capsule. So I'm going to go through this experiment. We're going to start here on the left. We have a mouse that was injected with this non-virulent, that means a strain that cannot cause disease, that did not have a capsule. Notice that we don't have a circle around these cells. That would represent a capsule. If we inject this mouse with this non-virulent strain of bacteria, that mouse will survive. It will basically kill that organism. If we did the same, but this time we used a capsulated strain, this strain is virulent because it has a capsule. That's what we're showing you right here. And because it's virulent, then it's going to cause disease in the mouse, and the mouse does die. Now, in both of these cases, the bacterial strain is live. What if we give the mouse a dead form of that virulent strain? That's this guy right here, but we're going to kill him first. By killing that bacteria is unable to replicate. It's unable to increase in numbers and cause disease. So therefore, the mouse will live. What if we give the mouse a combination of the dead virulent strain and the live non-virulent strain? Recall that in both cases, the mouse lives. Well, in this case, what happened is the mouse did not live. And when we actually sampled the mouse, we found that the living non-virulent strain became virulent because it acquired the genes from the dead virulent strain in order to allow it to make a capsule. By making a capsule, it now became virulent. The only way it could do this is if the dead bacteria released its DNA and it was picked up by the living bacteria, the non-virulent bacteria, through transformation. The other form of horizontal gene transfer is that of transduction. Now recall that viruses that infect bacteria, such as these bacteriophage, also have the ability to move short pieces of chromosomal DNA from one bacterial host cell to another in this process of transduction. Now we have generalized transduction and specialized transduction. In generalized transduction, any piece of the chromosomal DNA can be transferred to a new host by an accidental packaging of the chromosomal DNA into the phage head during phage assembly. And that's what we're seeing right here. This can only occur because as the bacterial cell is making hundreds and hundreds of bacteriophage, it's also shutting down because all of its energy is going towards the production of these virus particles that it's actually getting sick. And so in the process of shutting down and dying, it's going to fragment its chromosomal DNA into small pieces. As these bacteriophage are filling up their new capsid heads with virus DNA, some will accidentally be filled with bacterial host DNA instead. Now, whether these bacteriophage have virus DNA or bacterial DNA, they're still going to be able to go on and infect new host cells. If they happen to have bacterial DNA, the newly infected host cell will acquire that foreign bacterial DNA through this horizontal gene transfer. And that's what we're showing you right here. In contrast to generalized transduction, specialized transduction results from the imprecise excision or the removal of that prophage from the bacterial chromosome. Here's the prophage that has just been removed from the chromosome, but has a little purple. That is part of the chromosomal DNA that came with it. So it's going to carry that piece of bacterial chromosomal DNA that was flanking the edges of the prophage where it integrated inside the chromosome. So recall that only host DNA near that integration site can be carried over to a new host cell when we actually package the new capsid heads of those bacteriophage. Whether it's generalized transduction or specialized transduction, the newly infected bacterial host cells will acquire new properties from that previous host cell. In effect, they have the ability to express new traits that are passed on from the previous bacterial cells through this form of horizontal gene transfer. The third type of horizontal gene transfer is known as conjugation. In conjugation, DNA is directly transferred from one prokaryotic cell to another by means of a conjugation pilus, which brings the organisms into close contact with one another. In this case, the genes that encode for the ability to conjugate are located on a bacterial plasmid called the F plasmid. And the conjugation pilus is called the F pilus, or the conjugation pilus, or sometimes the sex pilus. This F plasmid genes will encode for both the proteins that are needed to make the pilus and those that are involved in replicating the F plasmid. Cells that contain the F plasmid and can make this pilus are called the F plus cells, or also the donor cells. And those lacking the plasmid are called the F minus cell, or the recipient cells. During a typical conjugation, the F pilus of the F plus cell comes into contact with the F minus cell, retracts the pilus, bringing the two cells in close contact with each other. A cytoplasmic bridge then forms between the two, and as the F plasmid is replicated in the F plus cell, a single strand of that DNA is going to be threaded through that bridge, and the recipient cell will start to make a complementary copy. So at the end, the recipient cell becomes an F plus cell with a complete F plasmid. Conjugation may also transfer chromosomal DNA, though. This is because the F plasmid occasionally integrates inside the bacterial chromosome, as shown here. So we start out with an F plus cell. The plasmid has become part of the chromosome, and as conjugation progresses and we start to replicate that plasmid, it's going to also replicate nearby host DNA. So similar to the imprecision removal of a prophage during specialized transduction, the integrated F plasmid can also be imprecisely removed from the chromosome, producing an F prime plasmid. This F prime plasmid will carry with it some host chromosomal DNA or bacterial chromosomal DNA that is adjacent to the integration site. Now, there are two major types of plasmids that are often associated with conjugation. They are these R plasmids that encode for antibiotic resistance, and there are the toxin plasmids. These plasmids have genes that allow for certain toxins to be made. So I'd like you to know that genes encoding for proteins that make a bacterial cell resistant to a particular antibiotic are known as R plasmids, and sometimes some R plasmids contain multiple genes that confer resistance to multiple antibiotics. This is a good way of increasing resistance to antibiotics through conjugation. Likewise, conjugation is a good way to transfer toxins that allow the organisms to be more pathogenic from one strain to another. The last form that I didn't mention in the previous slides, but this is an additional form of horizontal gene transfer, and that is of the use of transposons. This form is called transposition. Genetic elements that are called transposons are also called as jumping genes. They are molecules of DNA that include specialized inverted repeats. So I'm showing you an inverted repeat here in yellow and another one here in yellow. These transposons allow the entire sequence to be independently removed from one location of a chromosomal DNA and inserted into another chromosomal DNA. It could be at a different location of the same chromosome, or it can be from a different chromosome. Transposons may carry with them additional genes, moving these genes from one location to another with them. And for example, bacterial transposons can relocate antibiotic resistance genes, moving them from the chromosomes to the plasmids and vice versa. This mechanism has been shown to be responsible for the co-localization of multiple antibiotic resistance genes on a single R plasmid. Thus, transposition is another form of horizontal gene transfer. So with that will conclude this lecture on how asexual prokaryotes can achieve genetic diversity through the horizontal genetic transfer.